How deep can tissue cells feel? That is, how far into their surroundings can adherent cells sense a change in stiffness? I and my colleagues Emily Walton and Christopher Bruce, and our advisor, Professor Kristen Van Vliet, recently studied this question to learn about cells' mechanosensory abilities and the design of synthetic substrates for in vitro cell study. Many tissue cells are adherent, like these individual fibroblasts stretched out and growing on a flat polystyrene surface or substrate, but cells aren't uniformly sticky. Rather, they attach to their surroundings at discrete dynamic patches called focal adhesions. Tugging on these adhesion sites are cytoskeletal stress fibers of filamentous actin that hold the cell in a state of mechanical tension, the substrate underneath in compression. We can identify adhesion sites, which measure 1 to several microns large and are often located at the periphery of cells, by fluorescently staining for one of the participatory proteins, in this case, vinculin. At focal adhesions, the cell pulls at the adjacent substrate via transmembrane integrin molecules and senses the resulting deformation as a measure of the substrate's compliance. We idealize these regions as circular areas where uniform shear stress acts on the surface. Let's consider two possible substrate configurations. The first, a compliant semi-infinite region. The second, the same material in the form of a film atop an underlying, perfectly rigid base. This second configuration is common in laboratory experiments, for example when compliant hydrogels, used to mimic soft tissue, are supported by glass or plastic. In both configurations, the traction at the adhesion site deforms the surface. But for sufficiently thin films, the deformation is attenuated by the presence of the rigid base, which the cell begins to feel. The minimum thickness to avoid interference from the rigid base has been described as a critical thickness. But how exactly should we define this critical thickness? We found that multiple definitions were possible and in use. For example, the depth where one would find an arbitrarily small strain, say 0.1%, could be said to be a critical depth. Inserting a rigid underlying base at this depth would have little effect on surface movement. Or we might define a critical film thickness as one that maintains a certain surface deformation and thus sends a certain cue to adherent cells. A third definition might be the film thickness that attenuates surface deformation by a certain fraction, perhaps 1/10. Aren't these concepts all roughly equivalent? In fact, they're not. For example, these values change in different ways in response to changes in substrate stiffness. Stiffer substrates reduce the depth of strain propagation, but they require thicker films to obtain the same deformation. And the film thickness that cuts surface deformation by a certain fraction doesn't depend on film stiffness at all. To see why this is true, let's look closer at how these deformations are calculated. We use the Green's tensor, a 3x3 matrix that couples an applied force to the resulting displacement. In the case of surface deformation of a finite thickness film, the Green's tensor incorporates several Bessel functions and unique, intricate characteristic terms that are definitely best manipulated with a computer. Because we're interested in the response not from a single force but from a shear stress, we integrate the Green's tensor over the circular adhesion site area. Numerical calculations let us predict the deformation of an individual circular area, representing the adhesion site, for substrates of any stiffness. Not surprisingly, the site is motionless for very stiff substrates. After all, a cell can hardly deform glass, whose stiffness is 70 gigapascals, or polystyrene, about 1 gigapascal. But a cell can more easily deform hydrogels, whose stiffness can be in the kilopascals, as compliant as cells themselves, and 1,000 times more compliant than soft rubber. Let's look more closely at this deformation. The most obvious way to characterize surface motion is to ask, how far does the center of the circular area displace? But another, more subtle mode is the distortion, the compression of the leading edge and elongation of the trailing edge. these two modes turn out to be attenuated in slightly different ways. We bother to distinguish deformation modes because it's not yet known exactly how cells sense the mechanical properties of their environment. They're certainly sensing traction-induced deformation, but it's not clear whether they're sensing displacement, distortion, another mode, or a combination of these. For all deformation modes, however, a dimensionless number emerges in the equations. It's the ratio of the film thickness to the adhesion site radius. The appearance of this ratio alerted us to a deep connection between critical thickness and focal adhesion size. Let's consider two extremes in the value of this ratio. For the case of very thin films atop an underlying rigid base, all deformation is reduced to zero, as we expect. Conversely, when the film is very thick, the circular area displaces and distorts as if the cell were on a semi-infinite region, that is, as if no base existed. We numerically calculated the curves connecting these two extremes. Deformation is predicted to be affected, for example reduced by 1/10, when the compliant film thickness, the vertical distance to the rigid base, is about the same as focal adhesion size. We also predict that reductions in film thickness will be detected by the cell as increases in effective stiffness. After all, the only mechanical input to the cell is surface deformation, triggered by its own contraction. Note that material properties don't appear here. That's why we say that this measure of critical thickness is independent of substrate stiffness. We experimentally tested these predictions by seeding fibroblasts on compliant polyacrylamide gels of different thickness and using their spread area as an indication of mechanosensitive response. Co-author Chris Bruce prepared acrylamide solutions containing polystyrene spacer beads with diameters ranging from submicron to tens of microns. He compressed the films with a glass slide while they cured so that the bead size would determine the film thickness, and he adsorbed a layer of fibronectin protein to promote cell adhesion. We profiled scratches in the films with atomic force microscopy to determine their thickness, and also indented very thick films to determine the bulk stiffness, which was about 6 kilopascals for the recipe Chris used. When we seeded cells atop the films, we observed an immediate response to film thickness. Cells were more spread out on thinner films, which corresponds to how the same cells behave on stiffer films. The quantified spread cell area shows that cell morphology changes considerably when the films are on the order of 1 to 10 microns thick. We interpret this data as the cells sensing a larger effective stiffness when film thickness is reduced to the size of focal adhesions. Other groups have quantified how fibroblasts behave on thick polyacrylamide gels of various stiffness. When we calculate an effective stiffness by dividing the bulk stiffness of our gels by the numerical attenuation factor described earlier, we find that our data, replotted, corresponds well to these literature reports and thus confirms our predictions. In the old story of The Princess and the Pea, a mystery woman's royal identity is proven by her delicate reaction to a single pea hidden under a pile of mattresses. Individual adherent tissue cells also prove remarkably sensitive, responding strongly to changes in stiffness 1 to 10 microns beneath the surface upon which they attach. Our studies of cell-induced deformation are intended to support and advance both broad investigations of cell mechanosensitivity and, in a practical context, guidelines for synthetic substrate design. We're now looking for similar two-dimensional in vivo tissue configurations, such as the so-called basement membrane of blood vessels, so that we might apply our model to processes occurring in our bodies right now. I'm grateful to my co-authors and to the agencies that supported this work by providing research funding. This is John Maloney at the Laboratory for Material Chemomechanics at MIT.